Hello and welcome to Apologetics 101. I am your host, Brian Keith Bowen. All right, we've of course been doing the Genesis series lately, and I end up in interviewing. I have a special interview today. I have an interview with Dr. Jeffrey Tomkins, who's a geneticist from Institute for Creation Research, and it was such an honor to have him on my channel, and we had a great discussion on his method on his because he came up with an 84% number far as chimp and human relationship. He discovered that based on the papers and stuff like that that they wrote, that they were cherry picking the data a lot, and so a lot of evolutionists, and so he basically did his own evaluation of it and came up with a technique to eliminate the cherry picking so that it would come up with a more accurate number. And in recent times, this has really been attacked by a lot of critics. When I entered this interview, I didn't know what I was going to find. I didn't know. It's hard to really say with an interviewee because you don't know if they're going to tell you something that you agree with, if they're going to tell you something you don't agree with, if they're going to challenge your assumptions, if they're going to challenge other people's assumptions, if they're going to agree with mainstream views or go against those mainstream views. You don't really know what you're going to find until you actually get there and actually have a conversation with them. A lot of times I'm able to predict what they're going to say because I see what they write and other things, but in other cases you don't know. So I had no idea. I didn't know whether, I'm not a geneticist. A lot of the critics that was badgering me are not geneticists. Most of the criticisms about Tompkins' work is coming from people that are not geneticists. And so here was my chance to be able to talk to a real geneticist about this very discussion. And so, I didn't know what, going in, I didn't know what I was going to find. I didn't know if I was going to be proven right, proven wrong, if there was a flaw there, if there's a flaw here. I didn't know I was going to find. And that's typical of a lot of interviews. You don't really know that much about, what's going to happen. Or that I knew that there was critics who had issues with his methodology.
You are done, Tomkins. This method, it's through. It's finished. Throw the towel in.
I knew that there was some young Earth creationists even had uh, uh, questions about how his methodology would work under certain conditions.
No, it's just like I guess we could just go back to the fact that it would be nice if Tompkins would reply more, so we could get to the bottom of like what he's working on. He's very hard to contact.
And so I didn't know much of anything going into this discussion, but I did know at least two things. One, everyone wanted answers, and two, we're starting right now. Well, hello, and welcome to Apologetics
Hello.
101. I am your host, Christian apologist, Brian Keith Bowen. I have here today a geneticist from Institute for Creation Research, Dr. Jeffrey Tompkins. Um, and we're gonna go ahead and jump in. Dr. Tompkins, if you want to go ahead and introduce yourself and then show your I know you have a PowerPoint presentation. I have some questions here, but I'm going to save those until after you get done with your presentation. You might even answer a few of them. So take it away.
Okay, well, I got my PhD at Clemson University in 1996, and then after that, I did a postdoc at the Genome Center at Clemson University, and actually ended up becoming a faculty member there in the Department of Genetics and Biochemistry. And when the previous Genome Center director left for another job, they basically appointed me as the director of the Genomics Institute. And so I ran that for a number of years, and also taught classes in genetics and genomics and that sort of thing. And in 2009, I came to the Institute for Creation Research. So I had some friends here at ICR, and they finally talked me into leaving academia and coming to work for creation science full time. And so that's what I did. So I moved from Clemson to Dallas about 15 years ago, roughly. And so when I first came to ICR, they asked me to investigate the human chimpanzee DNA similarity issue. And so they basically said, you know, we go into churches and talk to people, and we're getting this, oh, humans and chimps are 98 to 99% similar in their DNA. What do you know about this? And can you come up with some answers? So I didn't really know anything at the time. I'd worked with a lot of different creatures and organisms, plants and animals and things, sequencing their DNA and looking at their genomes, but had never really compared human and chimp before. So I said, okay, I'll look into it. And it ended up being quite a quest. And that journey basically is what I'm going to be sharing in my talk today. So anyways, let's get into it. And let me pull up my slides. So let me share my screen. And get my presentation going. My PowerPoint okay?
Yeah, it's okay. I like your monkey.
So I call this monkey business in the chimp genome, and you'll basically see what I mean by this as we get going. And now my slides aren't advancing for some reason. There we go. Okay. So how did this whole idea of humans evolving from an ape get going to begin with? Well, really, Charles Darwin was the big impetus for this in the late 1800s. He proposed that humans evolved from apes. And of course, Darwin got this whole idea of a tree to Darwin. Life evolved from of course, Darwin didn't have all the biology and genetics that we have now, but basically creatures evolved over deep time and from a single-celled organism originally that somehow miraculously came into existence and then created the diversity of life that we see now in this tree-shaped pattern. We call it a phylogeny in the genetics business, but that's really not what we see in the real world. We see what myself and a number of other creation scientists would call a creation orchard. So we see lots of different kinds of creatures, plants, and animals. And we see variation within those kinds of creatures, but we don't really see one fundamental kind of creature or type of creature evolving into another one. And so here's just kind of a visual. So we see humans all over the earth, and there's a lot of variation among humans, and then dogs and other kinds of creatures. There's roses, many different forms of roses and things, but we never see anything evolving into really anything else, just a lot of variation within different kinds of creatures. So how did this whole idea in the modern era, anyways, of chimpanzees becoming the closest ancestor to humans get going? Well, we'll talk about that and what that means here as we move along. But I think it's safe to say that most modern scientists would claim that the chimpanzees are our closest living relative. And they largely do this based on the DNA sequence that they claim is more similar to human than some of the other great apes. And so a standard evolutionary tree that most modern evolutionists would claim would have humans up there at the top of the tree, and then chimpanzees, and then they would say we're more closely related after that to gorillas, orangutans, and then there's rhesus macaque, a monkey, as an outgroup there at the bottom. So that would be the standard phylogenetic tree for humans and their so-called living ancestors. But evolutionists can never really agree among themselves on most things. They always have debates. And so this was an interesting paper that's fairly recent, 2009. And actually, the authors of this paper claim that humans are more similar to orangutans than they are to chimps. And they did this based on the morphology or the anatomical characteristics that humans share with the various great apes. And so there isn't a complete consensus on this, but I would say based on the DNA, yet humans probably are more similar in their genome to chimpanzees. But the fact of the matter is that chimps, orangutans, humans, and gorillas, they're all just separate kinds of creatures. And they all have their unique similarities. And so humans kind of share, it's the mosaic. We share some traits with orangutans and some other traits with gorillas and some with chimps. So it's not really a clear-cut deal by any means. And in fact, the first thing that I discovered was this mosaic type of situation when I began investigating the molecular basis for human similarity with great apes. And so this is just a protein sequence, and proteins are chains of amino acids, and proteins are the workhorses in the cell. And so this is a protein called alpha motif protein 11, and I ran this in software that created a phylogenetic tree. And I did not get the standard evolutionary tree for this. In fact, gorillas for this particular protein were more similar to humans. And then there you have a rhesus macaque right below the gorilla, and then an orangutan and a chimp. So this particular biological sequence came up with a completely upside down or totally different tree than what evolutionists would claim. Here's another protein, ACAP3, and once again we get this mixed up picture, this mosaic is what I like to call it. And so it isn't a real clear-cut thing, and that's, like I said, that's because each kind of ape and humans they're their own created kinds of creatures. So when I began investigating the literature regarding the human chimp DNA similarity issue, which is really where you begin in any research project, you have to see what the literature is saying. This was one of the first papers that was published comparing human with a number of ape genomes. And what they did is they took the most similar sequences they could find between humans and great apes. So they cherry picked the data right up front, and then they ran it through software to construct evolutionary trees or phylogenetic trees. And this is a quote from the paper. So this was a study that was published where they compared the most identical sequences they could find between humans and great apes. So they cherry picked the data right up front. But anyways, they ran the DNA sequences into a software program that constructed evolutionary trees. And they said this in the paper: for about 23% of our genome, we share no immediate genetic ancestry with our closest living relative, the chimpanzee. So in other words, 23% of their evolutionary trees were all mixed up, just like I showed you in the previous slide. So there wasn't any clear pattern of human evolution from apes. It was a mosaic type situation. In 2012, they sequenced the gorilla genome, and they got the same results when they began constructing these phylogenetic trees with human DNA and the great apes. And they said in 30% of the genome, gorilla is closer to human or chimpanzee than the latter are to each other. So once again, a certain percentage of their evolutionary trees are not fitting the paradigm. They're indicating more of a mosaic sort of situation between the different genomes. Having said that, what is the actual DNA similarity between humans and chimpanzees? Is it 98 to 99% identical? Then I began tackling this issue. And what's interesting is that at the beginning of the chimpanzee genome project, which we're going to talk about in a little bit, they took 3 million sequences, DNA sequences at random from all over the chimp genome, and they compared those to the human genome. And this is a direct quote from the paper. They said about 2/3 of the, in other words, of the DNA, chimp DNA sequences, only 2/3 could be unambiguously aligned to DNA sequences in humans. So only 2/3 of the chimp DNA could they even match up accurately onto human. Well, that doesn't sound like a 98 to 99% similarity to me if they can only match up about 2/3 of the sequence. And so I began actually investigating all of the papers where they claimed a 98 to 99% DNA similarity. And there were 6 major papers that were published on that issue. And so I published a paper called Genomic Monkey Business, Estimates of Nearly Identical Human Chimp DNA Similarity Re-Evaluated Using Omitted Data. So I looked at all these 6 papers and where I could factor data back in that they had basically thrown out and discarded. I came up with actual similarities of 66 to 87%, depending on the paper. So obviously something was going on here. And in fact, what's interesting is that the first draft of the chimp genome, which was published in this 2005 paper, they didn't actually say what was the DNA similarity genome-wide. They just talked about the DNA similarity for the specific regions they could align and match up. And so that's what's kind of deceiving about this paper, is because those regions with the parameters on the algorithm they had seemed to indicate a very high similarity. But when I actually took the actual alignment similarity, multiplied it by the size of the chimp genome and the human genomes, factored those in, I came up with an 81% similarity maximum from that paper. But I had to do that basically going in and working with the data that was there, because they wouldn't admit this up front. So how have evolutionists achieved estimates of high similarity? Well, first of all, they've omitted the non-similar regions of the genome, which in some ways you can't fault them for that, because if the algorithm can't compare regions that are so dissimilar, or you have a region you're just not going to get any data because there's nothing to compare. And so there are large regions of the chip you just flat out can't match up because they're so different. And the one thing that I did notice was they were only comparing the highly similar segments, particularly the protein coding regions of the genome. They like to focus on those because those did kick out a high similarity. And so what do we call this? Well, I call it cherry picking the data. So in other words, these high estimates of DNA similarity, 98, 99%, were basically cherry picked data from only the most similar regions of the human and chimp genomes, with everything else being basically omitted. So let's talk about the human genome real quick, because it's important to keep this in the picture when we talk about how the chimp genome was sequenced, because there's a big difference between the two. So the human genome, the first draft was published in 2 separate journals, Science and Nature, because there was 2 separate consortiums. There was a private sector project led by Craig Venter, and then there was the public sector project led by Francis Collins, who was the head of the Human Genome Project at the time. And they agreed to You can see them shaking hands up there. They agreed to publish their genomes at essentially the same time. And so those were published in 2001, and those were that was the first rough draft of the human genome. And then the chimp genome was published in 2005 as far as the initial sequence and the initial draft of that. So what's the difference? Well, the human genome had billions of dollars of funding. There were labs all over the United States, Japan, Europe, really all over the world that were working on this project, billions of dollars in funding. And so they did a very methodical job of sequencing the human genome, but the chimpanzee genome project was very limited in its funding. And so they did not have near the amount of resources to sequence the chimp genome. And I'll explain why that is important. Well, let's talk about DNA sequencing. What is it? You know, a lot of people, they watch shows like Star Trek and they think, oh, you just stick something in the computer and out pops a genome. Well, it's not quite that simple. So let's talk about the Human Genome Sequencing Project. It was methodically done, as I said, and let's think about it with a good analogy here as a page-by-page sequencing strategy. So let's think about, if you will, a set of encyclopedias. So each encyclopedia would represent a chromosome, and then each page in that volume in the encyclopedia would represent a section of a chromosome. So in the Human Genome Project, they basically took a page-by-page sequencing strategy, very methodical, and because they had lots of money and lots of people working on it. Well, in the chimp genome, they did not have the advantage of all the funding and all the people and all the labs to work on the project. So they did what is called an all-at-once sequencing strategy. When I was in the business of genome sequencing, at Clemson, we called it shotgun. So think about taking the entire set of encyclopedias and throwing them all into a paper shredder all at once, and then trying to sort out all the little snippets that were produced and assign those to different volumes or in the set of encyclopedias. So let me show you how that would be done. So anyways, you would take the chimpanzee DNA sequences that were produced in such a manner where the entire genome was shotgun sequenced all at once. You would line those up with each other as best you could, and then you would assemble those onto a reference sequence. Well, what do you suppose the reference sequence was for chimp, or the scaffold as we would also call it? Well, it was human. In other words, they lined up all of the chimp sequences onto the human genome as a scaffold or a guide. And so as what they had then was various sections of DNA, of chimp DNA, lined up onto the human genome with a lot of gaps. So a good analogy as to how they put together the chimpanzee genome is this concept of how you would put together a jigsaw puzzle. So typically, most people that work jigsaw puzzles will look at the picture on the box, or there might be a little poster in the box that has a picture of the puzzle, which guides you to put all the pieces together. Well, that's how they put the chimp genome together, was based on the picture on the box, which was human, at random all over the genome. What's very interesting is that in 2018, a brand new DNA sequencing technology was employed that produced very long DNA sequence reads compared to the old technology. So this new technology would produce DNA snippets that were 30,000 bases long or more, compared to the old technology, which only produced DNA snippets 300 to say 1,000 bases long. And so this new technology was very effective in producing a high quality chimpanzee genome. And in fact, in this paper, what's very interesting is that the authors of the paper admitted that previous versions actually humanized. And so in this project, they did not use, initially anyways, the human genome as a scaffold to align all of the chimp sequences onto, because the sequences were so long they could basically assemble them on their own merits, and we call this a de novo assembly. And so, as what they said in the paper. Let me move this little thing here. They said the higher quality human genome assemblies, speaking of the previous ones, have often been used to guide the final stages of non-human genome projects, including the order and orientation of sequence contigs, and perhaps more importantly, the annotation of genes. That's an interesting statement, because I actually discovered they were inserting human genes into the chimpanzee genome electronically. They actually admitted this on a Genome Center website. And this is a very revealing comment that they also made. They said this bias has effectively humanized other ape genome assemblies. So that's quite an admission from the evolutionary community that previous versions of ape genomes were humanized because they lined them up onto the human genome. So anyways, this is another quote from the same author that was authored that previous paper, and he said in this separate paper, non-human primate genomes have not been finished to the same standard as the human reference genome and typically carry hundreds of thousands of gaps precisely over these regions of complex variation. So not only did they admit that the ape genomes had been humanized, but that also the quality of the ape genomes was not even close to being the same as human. But there's more problems going on than ape DNA being lined up onto the human genome and humanizing these ape genomes. Another problem is human DNA contamination in genomes that are out there in the public databases. And so this paper in 2011 was published, Abundant Human DNA Contamination Identified in Non-Primate Genome Databases. So they didn't even look at ape genomes, but they looked at bacteria and fish and all kinds of different genomes. They found that an entire section of a zebrafish chromosome was entirely human DNA. This is various animals besides apes, but they didn't even look at apes because I guess they figured out, oh, they're too close to human, so I guess we won't be able to figure out if there's contamination or not. Then in 2016, another paper was published called Human Contamination in Public Genome Assemblies, and once again they verified that the problem was still going on in 2016. So how do you get human DNA in all these databases that are not human? Well, people sneeze and they cough and there's DNA on their fingers. Even if they're wearing gloves, you can touch your face and get DNA on your gloves. There's DNA in the air floating around in the ventilation system. And so people in the early days of genome projects didn't realize what a problem that the human DNA contamination situation would be. So I've got some major questions, looking at all the literature that's been published and the various admissions that are being made about the state and the genome accurately assembled if it's put together based on the human genome. Another question I had, is human DNA contamination a problem in the chimpanzee genome? If it was, that would jack up the similarity as well. So what is the actual DNA similarity between humans and chimpanzees, and how can this be determined? So let's do an analogy here and talk about DNA similarity and what we actually mean by that. So here's a theoretical sequence of human DNA and then chimp DNA. And every time you see a vertical bar, that means there's a match. So where there's a T in human, there's a T in chimp, a C in human, a C in chimp, and so on. So this particular example there would be a 100% similarity. But let's say you have a C in human at one place and an A in chimp in another. And so that would be a mismatch, a single base mismatch. So in this example, 27 out of 28 bases match, and that would be a 96. One of the problems in DNA similarity as well is not the fact that we have a different base at a certain position in human versus chimp and vice versa, but that you also have these things called insertions and deletions, single base up to perhaps even hundreds to thousands in human compared to the chimp sequence or vice versa. So these insertions and deletions will decrease the similarity. So in this particular example, if we just counted the substitutions, we would have a 98% similarity. If we included the insertions and deletions with the substitutions, we would have an 81%. And so sometimes the algorithm can account for smaller insertions and deletions, but when they get too big, then the algorithm can't continue matching, it just breaks off. So I actually did a very large project where I looked at all 101 chimpanzee trace read data set. And I basically sampled 25,000 DNA sequences at random from each of the 100 software with a random sampling algorithm to do this. But anyways, I was able to create a data set of 2.5 million DNA sequences from chimp, 25,000 from each of the 101 data sets. So on average, these DNA sequences were about 700 bases long, and then I matched them onto human. And so I only recorded what I call the best hit or the longest match, because you'll get little snippets of DNA in a sequence kind of matching all over the place because of perhaps it's a little repetitive or whatever. So I would only record the best hit. And the nice thing about the Sanger style sequences that had a lot of what we call metadata associated with it. So there would be data with each individual sequence that told you when it was created, where it was created, what lab it was created in, and so forth. So I was able to create a relational database and with this metadata, and also combine it with the DNA sequence similarity for each. Map these out over time, and I saw a very interesting trend. And the trend that I saw was the DNA sequence similarity between human and chimp drastically declined over time. So what's going on? I think there was human DNA contamination at the beginning of the chimp genome project. So just to simplify this data, between the years 2002 and 2004, the overall DNA similarity for these reads with human was 92%. For each, on average, 92% was the average DNA sequence identity for these reads. But during the last phase of the chimp genome project, 2005 to 2011, the DNA similarity on average was only 85%. So why the 7% difference? So what's interesting is those years, 2002 to 2004, were actually the years that went into the draft genome of the chimp project that I showed you that was published in 2005. So I think that in the early phases of the chimp genome project, there's a lot of human DNA contamination in the data sets. That's why they were really a whopping 7% more similar to human than the sequences at the latter end of the chimp genome project. Contamination, in my opinion, well, that presents a huge problem to the sequences being lined up onto the human genome as a scaffold. Chimp genome sequence that was put out in 2018. It was a new technology. And so I had 18,000 segments at random from around the chimp genome that I lined up onto the human genome. Those DNA sequences were on average about 30,000 bases long. But of those 30,000 base chunks, I could only line up 10,000 bases on average. So in other words, only a third of those 30,000 base chunks of chimp DNA would even align to human on average. And the average alignment identity or similarity that I achieved from those was 84%. So basically the chimp genome at a maximum is only 84% similar to human and probably less than that. So what's really interesting is that Richard Buggs, professor of evolutionary genomics at the University of London, Queen Mary, actually did a study himself where he downloaded a DNA sequence that was lined up, chimp DNA sequence that was lined up onto the human genome using a different algorithm than the one that I used. And he said this in a blog that he posted: the percentage of nucleotides in the human genome that had one-to-one exact matches essentially that I came up with. So what's the evolutionary problem with 84%? Some people would say, well, that sounds pretty similar, even if it's not 98, 99%. Well, the problem is that theoretical evolutionists who are trying to explain human evolution from a chimp ancestor or an ancestor we shared with chimp need a 98 to 99% genome similarity to explain human evolution with the known mutation rates that they've actually determined in humans and chimps in 3 to 6 million years since we supposedly shared a common ancestor with chimps. So for theoretical human evolution from a common ancestor with a chimp to even work, you need something close to a 99% similarity. So 84% obviously totally kicks that out the door. So this whole issue of the human genome supposedly evolving from a common ancestor with chimp really begs the question, are genomes really evolving and getting better? Are they gaining information and getting better, or are they actually devolving and losing information? Well, common sense would tell you that they're devolving. You know, even computer systems become corrupt over time. The data on your hard drive needs to be repaired and fixed. And so essentially the human genome is not evolving and getting better. It's actually devolving over time. So this whole idea of evolution has serious problems fundamentally to begin with. In fact, let's look biblically at the lifespans of humans, and we can see that since the flood, the global flood, about 4,300 years ago, that human lifespans have been decreasing significantly. And so John Sanford and his colleagues have actually been analyzing that and actually proving that is the case. But what's really interesting is that several papers were published, secular groups, where they actually looked at mutations in what's called rare coding variation. In other words, they looked at the mutations in protein coding segments of the human genome, and we call those exomes. And so one of these papers was Evolution and Functional Impact of Rare Coding Variation from Deep Sequencing of Human Exomes. And the other paper was similar, and they literally sequenced thousands of human exomes, just the protein coding regions, from humans all over North America and Europe. And they, instead of calibrating their models with deep evolutionary time, they actually took a very realistic empirical approach where they looked at population demographics. In other words, how fast populations actually grow from the data that we have all around the world. And they actually applied a real empirical model of population demographics to their data they were extracting from sequencing the exomes. And this is a graph from one of the papers where it shows that human variation only goes back about 6,000 years. Well, that's really interesting. And in fact, these rare variants were associated with mutations in the exome that were largely associated with heart disease, diabetes, all sorts of bad stuff. In fact, 80% or more of these mutations were not doing anything good to the human genome. They were associated with some sort of human health issue. Needless to say, though, that variation based on their empirical models in both of these papers only went back about 6,000 years. Well, biblically speaking, what happened 6,000 years ago? Well, humans were created about 6,000 years ago. If you look at the chronologies and genealogies in the Bible, in the Hebrew text, we come up with a creation of about 6,000 years ago. And it's really interesting. That's what we see with this data. So right there, about 6,000 years ago, and you can see human variation really begins taking off. This mutation-based variation about 5,000 years ago, which was when the global flood was and the Earth was repopulated with Noah's 3 sons and their wives. So what about this whole idea of human evolution anyways? I just want to go over a few slides as to why that's really bogus from an anthropology perspective. So this is typically a typical figure that you might see. And basically you see apes there at the start. You see humans there at the end. But essentially everything in between is hypothetical and theoretical. That's all we really see in the fossil record anyways, is humans and apes. So let's start with Lucy. So commonly known as Australopithecus afarensis. The skeleton was only about 40% complete. That's all they had of it, and it was discovered in 1974. They dated Lucy at about 3 million years old and claimed that she was bipedal and walked upright. However, we've got major problems with this, admitted by the evolutionists themselves. Did Lucy really walk upright? So Eric Stockstad, an evolutionist, published this article in Science in 2000, and he said, I walked over to the cabinet, pulled out Lucy, and shazam, she had the morphology that was classic for knuckle walkers. So in other words, when Lucy wasn't swinging around in trees, she was on the ground, walking around on all fours like chimpanzees do, because they're designed to be knuckle walkers. They have special locking wrists that humans don't have, so they can walk around and do that. And there's a picture of a chimp doing what chimps do, knuckle walking when they're on the ground. And so this is another quote from an evolutionist. I love the name of this article, Lucy on the Ground with Knuckles. And this was published in Science News in 2000. And they said Australopithecus anamempsis and skeleton known as Lucy had wrists capable of locking the hands in place during knuckle walking. Walking Ancestor Nature 2000. And these evolutionists said, regardless of the status of Lucy's knee joint, which they can never come to a conclusion on, new evidence has come forth that Lucy had the morphology of a knuckle walker. And this is a great quote from 2016, and the title of this paper says it all, really. From Australopithecus to Homo, the transition that wasn't. So they said in the paper, although the transition from Australopithecus to Homo is usually thought of as a momentous transformation, the fossil record bearing on the origin and earliest evolution of Homo is virtually undocumented. And that was in the Royal Society 2016, and nothing has changed since then. So what about this whole idea of Neanderthals and so-called primitive archaic traits in humans? The fact of the matter is you wouldn't even know a Neanderthal unless he came up to you with a name tag and a spear on the street, because they looked human. And in fact, now they're sequencing human DNA, human DNA. They're sequencing DNA from Neanderthals that have been actually ritually buried in caves because they're human. And so we see stories like this in the news quite frequently now. Humans bred with this mysterious species more than once, new study shows. your Neanderthal DNA might actually be doing you some good. So yes, there are evolutionists out there saying we need to clone Neanderthal brain genes and put them in our own genome because Neanderthals had bigger brains. So that doesn't sound like evolutionary thinking to me. Ancient humans may have been mothers to some Neanderthals earlier than we thought. And so this is the general consensus that humans and Neanderthals interbred, and that essentially Neanderthals, according to evolutionists, were archaic humans. But the fact of the matter is they were just humans. So the Neanderthal skull was typically larger than a modern human. They had a sloping forehead and a enhanced brow ridge, or pronounced brow ridge. And in fact, the Homo erectus skulls, which is another class of archaic human, their skulls were not as large as Neanderthal skulls. They were a bit smaller, but they had essentially the same feature, the sloping forehead, the large brow ridge. But is this really an archaic trait? So there is a dentist in South Africa who takes pictures, who's a Christian and a creationist, takes pictures of his patients that have these so-called archaic traits. And here's a guy that was in, I don't know if he's having a cavity filled or what, but anyways, he got his picture taken. Look at the brow ridge on this guy, and he is a young man and alive and well. I took these pictures of rugby players in Eastern Europe, which it's interesting because Neanderthals are typically found in Eastern Europe, also in other parts of the Middle East and some other places. But anyways, look at these guys. They have sloping foreheads and big brow ridges. This is a famous guy, Nikolai Valuev, a former professional boxer. He's a member of the Russian parliament now. He has a sloping forehead and a huge brow ridge as well. So this really isn't an ancient archaic trait. It's actually found among modern humans today in different parts of the world. But what about this whole idea of a sagittal crest, or this ridge along the top of some people's heads that evolutionists once claimed was archaic? And so the arrow is pointing to a guy there with a pronounced sagittal crest, but the guy up on in the suit there is a famous neurosurgeon, and he has a sagittal crest, and he's a pretty smart guy. And then of course the other guy over there, he pilots a star cruiser across the galaxy, so he must be pretty smart. But the fact of the matter is these so-called archaic traits are really found among modern humans. There's just a lot of variation among humans. So here's two professional tennis players. This was actually a very close match, but the one guy is almost 7 feet tall and the other guy is 5'7". So anyways, there's just a lot of variation among humans for different traits. And it's variation, not evolution. All the other animals that God created, humans were created in God's image. So the Bible says, so God created man in his own image. In the image of God, he created him, male and female, he created them. So humans were the pinnacle of creation and created in God's image. And this is why, in my opinion, human evolution is one of the top evolutionary dogmas, because they're really attacking Genesis and the nature of man fundamentally at the beginning of the scriptures. So why is this important? Well, it has to do with the idea of a historical Adam. Did Adam really exist, or did we evolve from apes? And of course, there's an organization that is theistic evolution at its core. It's biologos. And you might as well be on the Berkeley University website reading about evolution, because you'll get the same story. This is a problem, because the Bible has very distinct and clear genealogies and chronologies back to the original Adam. Why is that important? It was brought into the world through Adam and Eve when they fell at the beginning in Genesis, and that's documented in Genesis chapter 3. But the Apostle Paul clarified the importance of this in his epistles. In Romans 5:12, he says, therefore, just as through one man sin entered the world, and death through sin, and thus death spread to all men because all sin. So in other words, through one man, meaning Adam, sin entered the world. 1 Corinthians 15:22, for as in Adam all die, even so in Christ. And all this evil come from, but why is creation in the state that it's in? And it's because important for explaining why do we find all this evil in the world, and why are humans so evil? I actually have a book called Chimps and Humans: A Geneticist Discovers DNA Evidence That Challenges Evolution, and I talk about the human chimp DNA similarity in this book. I talk about other important aspects of the whole paradigm, the so-called chromosome 2 fusion, pseudogenes, junk DNA, everything that you could imagine that relates to the human chimp DNA issue is in this book. It was just published about a year ago, so it's pretty much hot off the press. And I have another book, The Design and Complexity of the Cell. It's kind of an intelligent design approach to describing the complexity of the cell and how that could not have come about anyway whatsoever through evolution, because cells are incredibly complex. And so anyways, so I have those two books that are available. If you're interested, you can go to icr.org and get those, or they're also on Amazon.
All right, awesome. That was great presentation. I do plan on doing some videos later on Australopithecus and things like Neanderthals and things like that. So I appreciate that. But today we're primarily going to be focused on your particular methodology. And I have some questions because I don't think I heard anything on chromosome 2 fusion in your presentation. Could you explain that real quick before we jump into the other stuff?
Yeah, I actually have done entire presentations on that can get very technical. So in the so-called chromosome 2 fusion, evolutionists believe that two chromosomes fused from the ape genome to form human chromosome number 2. The reason they had to go that route was because humans have a chromosome number, a diploid number of 46 chromosomes, and apes have 48 chromosomes. And so they did find some gross similarities doing cytogenetic spreads of human and ape chromosomes. But they also found some problems with it as well. But anyway, so that's what led to the whole idea of it. And then they thought they found in the early 1990s actually a DNA sequence that represented the fusion site, which they claimed was a head-to-head fusion of human telomeres. And so it was a very small signature. It was only about 800 bases long. And the reason I say small is because human chromosomes are 5,000 to 15, or I'm sorry, their telomeres at the ends of them are 5,000 to 15,000 bases long. So if you had two telomeres allegedly fused together, you should have a signature of 10,000 to 30,000 bases, not 800. And so on top of that, the fusion site is very muddled, if you will. It's evolutionists would call it degenerate. In other words, it's not a pristine head-to-head fusion. It's only about 70%, similar to a pristine fusion of the same size, about 800 bases. So anyway, so it isn't that clear cut. And on top of that, the fusion site is actually found inside a long non-coding RNA gene. And if things couldn't get any worse for the evolutionary fusion story, the fusion site is actually a second promoter or a switch inside this gene. It binds RNA polymerase 2, which initiates transcription in the middle of the fusion, so-called fusion site. All sorts of transcription factors bind to the fusion site. And I basically mined all these ENCODE data sets to verify all of this. And if you look on a genome browser, you will see the fusion site inside at the beginning of this gene. So many genes in the human genome have multiple promoters or these switches. A lot of these switches are in front of the gene, and a lot of the switches are inside the gene right at the beginning, maybe after the first exon, like the fusion site is. So anyways, it is actually a functional feature inside a gene. And this gene is an important gene. I actually looked at the expression of this gene. It's expressed in many different tissues in the human body. It's connected with the expression of other genes in top-level networks, including the formation of blood cells. And I actually annotated the messenger RNAs that were produced from this particular gene that the fusion site is in. And these messenger RNAs are full of all sorts of RNA binding sites and all sorts of functional features. So it's not an evolution who might say, oh, it's just spurious transcription, you know? No, it's a real gene that's producing real transcripts that do real stuff, important stuff. And so the fusion site is bogus. It is a feature, a functional feature inside a gene. And so I can get really deep in the technical muck with this. And I actually have an entire talk that I gave at the International Conference on Creation in 2018, where I actually developed some software to scan the human genome for internal telomere sequences and then line up those genome coordinates with all these all sorts of internal telomere-like sequences that are associated with a variety of functional aspects and all sorts of things. So anyways, and I actually published a paper on that. The whole fusion scenario is not viable when you begin looking at the genetic data. In fact, I even looked at the so-called cryptic centromere site, which is less convincing than the fusion site. So if you had two chromosomes fuse, because each chromosome has its own centromere, theoretically you would get two centromeres, and then one of them would supposedly be deactivated after the fusion. So I actually looked at the chromosome 2 cryptic centromere site, and it was also inside a gene. In fact, it was in a huge protein coding gene that produced a transmembrane spanning protein that after protein is produced to be transported to the cell membrane and work as some sort of a receptor with the part of the protein sticking outside the cell, the other part sticking inside the cell. And so any was actually inside this large protein coding gene. And in fact, part of the so-called cryptic centromere actually coded for the actual resulting protein from the processed messenger RNA after it was transcribed. So, and on top of that, this so-called cryptic centromere actually had human-specific repetitive elements in it that you don't even find in apes. And so the whole cryptic centromere site thing was even as problematic as the so-called fusion site. So any papers on that, maybe you could put some links up to those. I can send you those links later.
Yes,
So in a nutshell, that's
yes, I'm gonna link all your peer review papers that I find on Answers Research Journal. I'm gonna link those in the description box. I have your book right here, and you do make an excellent case against the idea of fusion. You mentioned the 798 base pair length, which is really small compared to where it should be. Also, you got end-to-end fusion. You mentioned that as well in your book with telomere and telomere, which doesn't happen. Is that correct?
yeah. The only time we actually see telomere fusions is in cancer cells, which are basically gone hog wild and are not healthy. We do not see actual telomere fusions in nature. We see other types of fusions, but they don't involve two telomeres coming together.
Right.
And so even if we look at what happens in nature, that doesn't help the story either. And of course, chromosome fusions in nature are pretty rare. They have happened on occasion, but they are pretty rare. But they do not involve two telomeres fusing together. Sometimes they involve a telomere, but the other side of the equation is not a telomere. It's another type of DNA sequence. And in
Right.
fact, I actually published a paper on human telomeres and showed how well designed they are. So the DNA at the ends of chromosomes, these telomeres folds back into itself, and it's covered and protected with a protein complex with multiple proteins in it called the shelterin complex, which not only protects the ends of the chromosomes from fusing, but it's also a dynamic machine, if you will, that interacts with the cell cycle, and it assists in cell division in various aspects of that. And so it's actually the ends of chromosomes are actually very, very well protected and very vital to the healthy function of a cell. And so they're not designed to just fuse. They're not just hanging out there waiting to fuse.
As far as the genes inside, though, you said it was inside a gene. Um, does do evolutionists think this is a pseudogene that it's inside? And also, what is a pseudogene? You talked about that in your book.
Yeah, so most of the pseudogenes, the so-called pseudogenes, are long non-coding RNA in the genome. But most of them now that they have analyzed are functional and important to human health, of health defects in humans. So actually, pseudogenes, or more properly, long non-coding RNA genes, are really hot research items in human health in the biomedical field right now. In fact, they have found that there are twice as many long non-coding RNA genes than there are protein coding genes in the human genome that are just as important as the protein coding genes. And so the human genome is full of all of these genes that are doing functionally important things. Some of them stay in the nucleus. Some of them go into the cytoplasm and do things. Some of them are even exported outside the cell. It's important, and they're really a hot research field right now out there in the biomedical research field in humans.
What about junk DNA? What they call junk DNA? What exactly is that? And is there really any such thing as junk DNA?
No, junk DNA, actually, people don't realize this, but it was derived from what was called the neutral model theory of evolution, where supposedly vast regions of the human genome were not functional and they were free to evolve and create and spit out new genes. And then as they began analyzing the human genome more, they found out that much of it was not protein coding, and they just flat out didn't know what it did. Well, just because you don't know what something does not mean it's not functional. And so the Human ENCODE project, which stands for Encyclopedia of DNA Elements, when they began really sequencing the human genome, that's junk DNA. It's not coding for anything. It's not doing anything. It's just free to evolve. Weren't really buying that. They were thinking, well, I think there's a lot of stuff that's functional there, but we just don't know what it does. So massive amounts of funding. The first round of papers that were published by the ENCODE project were in 2012 in all the top journals. And the general consensus now is that almost the entire human genome is functional. It's doing something. In fact, one paper was published by an ENCODE scientist, and he said the human genome is an RNA machine. In other words, the entire human genome they discovered was transcribed into RNA from almost end to end. The chromosomes in the human genome and animal genomes are painted with this RNA matrix that literally creates a sort of functionality. We're not sure exactly what's going on, but that is necessary for proper chromosome structure and function. And so the genome really is very complex. The 3D structure of the human genome is different in every organ and even across organs, because there's different cell types, not only between organs, but within organs as well in the human body. And every cell has its own unique 3D structure, which is required for the specific type of cell that it is and what that cell is doing and the kind of gene expression that is going on inside that cell. And so it turns out that RNA is a major player in creating genome structure, genome functionality. We're just barely beginning to discover what is going on with RNA in the genome. But one thing that we know is that the human genome is literally transcribed, turned into RNA from almost end to end. And it's critical and it's important to good health.
All right. I got one more question on junk DNA. When you researched the cherry picking, did you find that the junk DNA was among the things that they was excluding from those numbers?
I could not detect that because they didn't necessarily distinguish that in a lot of cases. But I suspect that that long non-coding RNAs, these intergenic regions between protein coding genes, even regions within genes that did not necessarily directly code for end up in the sequence of a protein or a mature messenger RNA transcript that was translated into a protein. I do suspect that a lot of those regions were not represented in their analyses.
Awesome. This discussion has been a huge event going on YouTube, and people have been talking about your method. People's been talking about your chromosome, the alleged chromosome 2 fusion claimed by evolutionists, and there's people on both sides. They're debating the subject back and forth, and so it's been a very big controversy there. And when you think that that the biggest thing that the evolutionists would criticize and say no, we're not doing that, was be all the cherry picking that you've accused them of. But instead what they've done is latched on to your particular methodology, where they challenge whether or not if your method can get this correct number. So we're going to deal with some criticisms. The first one is what I like to call the math argument, because it's one where, before I do that, I need to clarify something. The gap and ungap, that means with including indels, which are insertion and deletions, or without insertion and deletions. One of the criticisms was on your first paper didn't include gaps. Is that correct? Or the other way?
There was an initial paper I did, I think back in 2013, where I did not include gapping in the algorithm. And it turns out the algorithm was bad anyways, because it was kicking out sequences in large data sets. And I actually talk about that in my later paper. So it's not like I was, not discussing the issues that were going on later on. So yeah, people that want to be naysayers and so forth will go after something you did originally that, and not acknowledge the most recent work you did. And fundamentally, people have presuppositions. Some people are bound and determined to deny there is a God. They don't care what evidence you give them whatsoever, and then they want to nitpick something that really doesn't. It's kind of an esoteric angle that really doesn't invalidate what you really discovered later on. you know, um, anyways, in fact, there were problems with that algorithm. I reported those to the NIH, who created the algorithm, and I was actually corresponding with the the programmers there at the NIH and sending them data sets because I was trying to convince them, hey, your algorithm is messed up. It's kicking out data. And I finally was able to do that. I actually had to send them data. I had to send them results from analyses, and they did fix the algorithm. And so in later papers, I did use gapping, and the algorithm was behaving itself and was not kicking out data. And so, yeah,
So your
yeah,
initial
people
paper,
will people will focus, on an earlier paper, and then you produce research later on, and they ignore it. So,
right?
I mean, what can you do?
Right. So your
And the
initial,
fact of the matter,
oh,
yeah, go ahead.
sorry. I was gonna say your so initially some earlier ones it was ungapped because of the fact that you had a bug in that particular algorithm, but then that problem got fixed and
Well, no,
resolved.
I was using an ungapped algorithm because actually a lot of evolutionists were using ungapped algorithms in the early days as well.
Okay,
So
that's
actually this particular algorithm has been greatly improved over the years. And so you just got to use the tools that you have at the time. So yeah, the most recent paper I did, I used the most liberal, gapping that I could on the algorithm. And yeah, people will throw out anything to discredit you. It doesn't matter what it is. So
right. Some people say that they say yeah, but even though he switched back to gap, the it was unweighted. What does that mean exactly when they say that they don't think you weighted your sequence?
takes into account the weighting.
Could you explain that?
You can't set the weight really of the algorithm. You can statistically you can set the gap extension. I don't know what people are talking about. I mean, it's nonsense. I did what I could to create a situation where the algorithm could get over at least the small gaps. The algorithm can't get over the big gaps. Yeah, why aren't people talking about, oh, why could he only align 30% of these 30,000 base chunks of DNA with the most liberal gapping he could? And no one's talking about, the 2/3 of the DNA sequence that the algorithm, couldn't even align because the difference got so extreme, the algorithm had to break off its alignment.
Does this
Why aren't
affect your
guys?
does this affect your number any, whether it's weighted or unweighted?
Like I said, the weighting is included in the setting of the parameters of the algorithm.
So in other words, it's automatically set within the algorithm itself.
Yeah, you set the statistical threshold, you set the gapping parameter. It's yeah.
Also,
It like I say, people don't care what you discover. I mean, I could discover 50% DNA similarity or whatever. It doesn't matter. And unless the Holy Spirit is really working on that person to reveal the truth to them, apprehend the truth. And I would suggest to people out there, do what I did before I became a Christian. I was driving down the road in my car and I said, God, if you are out there, please do something. Reveal yourself to me. I'm tired of living life the way I am.
uh, I'm gonna show a clip. Now, I think they're trying to think that some people will try to take all your research in a particular paper. They say that they're like and put it in and they'll put the numbers in an Excel type document, and then they'll
Well,
set their
I can tell you what people are talking about. So there is a guy in Australia. I don't know if he's still alive because he got cancer actually a couple years ago. Um, he is a computer programmer, and he literally attacks creationists. It doesn't matter whether they're talking about human chimp DNA or whatever. So anyways, this guy's a computer programmer, and him and some other guys were working for a financial trading company, and they started their own company and became millionaires literally overnight. About all this money he made and how he was going to spend his life now attacking me and other creationists. So this is the guy that put out that video, and it's utterly bogus from start to finish. And yeah, it's I know what you're talking about.
You're speaking of
He's a
Glenn.
nefarious character,
I know
basically.
who you're talking about, Glenn Williamson, right?
Glenn Williamson, yeah. He calls himself Ruhif, and I'm not the only one he's attacked. He will literally use email addresses in the creation science. He's attacked multiple people in our ministry, at other ministries. Yet even if you're just an intelligent design person, you know, you don't even necessarily believe in a biblical timeline or whatever, he will figure out who you are and attack you one way or another. But one interesting point he made at the very beginning of his video is he said, "Oh, well, Tompkins just determined the average of these DNA similarities for all these alignments." And he said, "Here's an example. Let's say you drove here and it was only, I forget what he said, maybe 20 miles, but then you drove to this other part of Australia and it was 1,000 miles, and then you took the average. Well, what's that? That's a sample size of 2." What was my sample size? 18,000, you know? And so statistically, that his argument was nonsense. And then he never explained all of the sequences that had super low similarities. And he also only looked at the alignment similarities, ignored the 2/3 of the sequence that didn't even align. And so my paper was totally accurate. The average identity of those 18,000, average, the mean of those 18,000 sequences was 84%. And so he went from all these weird, stupid angles. Some of them were just totally bogus. And so yeah, that's the video that's out there. And anyways, so I'm not the only target of this guy.
Right. One of the things I found out,
other people have been attacked as well.
Yeah, one of the things I found
What's
out is
that?
that all of these critics seem to be bouncing off of each other, like they're using each other's arguments, and they're just kind of
Oh,
almost
right.
absorbing each other. I know that like, you know, you have Williamson or Williamson, the Glenn Williamson, and he said he got his information from Dr. Joshua Swamidas and
Oh,
Erika.
Swamidas is another. Yeah, let me mention Swamidas.
All right.
So basically, this one study that I did, it literally took 6 months of time on 2 servers, each one having 40 logical cores and 384 gigabytes of RAM. Last study that I did, and Swamidas claims he went home from work one night and repeated my study in a couple hours. And I'm like, are you kidding me? You're a flat out liar. It's just nonsense. And I wished I'd have saved that quote. He put it up on his blog, and I'm thinking, that's just a bold-faced lie. You didn't go home and one night on your little PC and repeat a study that took me, 6 months to do on these powerful servers. It's nonsense. And so, yeah, I think, yeah, Swami Das, and who's supposed to be a Christian, and this Glenn Williamson, who's obviously a complete and total pagan. Why are these guys collaborating?
Yeah, and there was a video just posted a few days ago by a evolutionist named Erika from Gutsy Gibbon, and she actually posted a video where she was trying to target that, and she used a lot of these same arguments. She used the arguments from math, and she used the arguments from, oh, she focused on, the bug in the original system or whatever the case may be, as the algorithm. And she said she'd done these tests, these experiments, and she also had her regular computer system. She said she used BLAST. She said, and she said that she did a study on first. The first one was on the things that we would agree is the same kind of animal. And she said that she, come up with all these differences and stuff like that. Now, of course, that's easily explainable. I mean, we don't expect them to be exactly alike anyhow. But then she also said that she also tested it against itself, like using your method, and she said that she didn't get 100% identity. Now, of course, Swami Das said the same thing on his Peaceful Science blog, like you mentioned, where he tried to test rats and mice. He claimed. Again, to me, I'm already automatically skeptical of this because they're using their personal computer. Granted, they're using some similar algorithms, but they're using the personal computer. She's claiming that she did it exactly as it's spelled out in the papers, what she's claiming. But I read the methods in some of your papers and they're pretty advanced. So I'm sitting there thinking, okay, it's kind of hard for me to believe that they're set up exactly in a laboratory situation where they will be able to actually cover this in there. I know she if she's watching this, which she will be, she probably get offended. Oh yeah, I did do it exactly as it was spelled out in the paper. But she's not. Again, it's the whole. I don't know if she's set up with all the right equipment, all the right gear, and even whether she even did your method perfectly, regardless what she said she did. But any given case, how would you respond to people saying that they've used this experiment, that they've tested it on similar kinds, that they've tested against itself, the same kind, not just the same kind of animal, but like test my DNA with my DNA, and they say that okay, they pounce on it, they say nope, the method doesn't work. I mean, how do you respond to people like that wants to claim that they've done these types of experiments? Now, granted, like you said, it took you a while to actually do the experiments, and you got resources, you got much more powerful stuff. But how do you respond to people who said that they've basically tried to replicate this back at home, and they say your methodology doesn't work?
Well, right off the top of my head, I don't believe him because I would basically log in to a Linux server and start these jobs with shell scripts running and then log out, and the jobs would just keep running for weeks because the data sets were so big. And so I know Swami Das, what he said was utter nonsense when I first read that. And I almost felt like sending the guy an email, but I didn't, calling him out on it because it was so ridiculous. I have no idea what this gutsick Gibbon girl did. But yeah, you're right. They just kind of feed off of each other and put out all this stuff on the internet. I seriously doubt any of them have come close to replicating what I did because I know the amount of time and effort and processing power it took. And to think these people went home and did it at their house is, is crazy. Unless they had access to really powerful servers like I do. So right off the top of my head, I know Swami Das was utter nonsense what he said, and yeah, and I know that Glenn Williamson, the stuff he said was very misleading and very caustic, and but I don't have time to I feel like sometimes I'm in a whack-a-mole type situation where someone pops up and says this and that, and even though it's just utter nonsense, someone else is popping up over there and someone over there and someone over there. And before I know it, I'm spending all my time trying to respond, to people that they're creating all these diversions when I could be actually doing real research instead of, doing this whack-a-mole kind of thing. So,
I feel like that's my life sometimes.
I only have so much time in the day. And, like I say, there's going to be people out there that are so caustic and anti-God that they'll do and say anything. Like, I think a good example is and I didn't watch this debate, but James Tour, who is kind of an origin of life specialist, recently did a debate, and the guy he debated, just was completely obnoxious and threw out all these ad hominem attacks and this and that, and it was just a circus. And so those are the people that are out there. It doesn't matter what you do, what kind of data you produce, they're going to come up with some sort of a diversion. And they, like you said, that's a great comment. Yeah, they all feed off of each other, and before you know it, they're creating all this nonsense out on the internet. And I can only I've only got so many hours in the day, so yeah.
I do have a question though. If your methodology was applied correctly using the same system, the same tools and resources, if you did it yourself and just kind of apply it on, one on same kind animals versus if you apply it like if they're exact copies of each other, would you get? I mean, do you believe you would get accurate numbers regarding that? And two, would you actually do that? I mean, that sounds like a fun project. It sounds like something that will be a good way to actually see just how you compare those things. I don't know if do you think that other kinds of comparisons have been cherry picked?
The thing is that when we talk about the human genome, it is the most heavily funded genome project in the world, because we want to get to the bottom of why do we have all these health problems and diseases and things. So they have actually sequenced human genomes all over the globe ad nauseum. And so I don't know what these people are doing, but the fact of the matter is that based on single base variations, we call them single nucleotide polymorphisms, or SNPs. And you can pull up any genome browser and all the SNPs are listed in there for the human genome. But if we just include the SNPs, or these single base differences, any two human genomes can be about 99% similar. Actually, I think the figure is 99 point something percent similar. But if you include the structural variance in between humans, any two human genomes can be 4 to 4.5% different from each other if you include what's called structural variants, and which they are actually now doing, because the DNA sequencing technology with the PacBio sequencing, the ion torrent, and all these new technologies are actually able to sequence through the structural variants in the human genome. And so if you include those, any two human genomes can be up to 4 to 4.5% different from each other. Well, if we have 4.5% difference from each other potentially, how are we only 2% different from a chimp? It's nonsense. And these people that claim, oh, I discovered this by comparing the same genome. I mean, you know what? You're the entire global human genomics community has already done it. You can just go to the literature and see what they did.
All right.
So, anyways, and they found pretty much the same situation among populations of chimps that they've sequenced as well. So I don't know what these people are doing, but yeah, as far as comparing one human genome to another, it's been done ad nauseum all over the world in the Human Genome Project, which has been going on since early 1990s.
Right. I do have Erika from Guts at Gibbon was actually in the live chat when you did your interview with Christopher Roop. or what his name is,
Yeah.
Chris. Yeah, that was her. She did give me a question to ask you though. I quoted it exactly. This is the question that she has verbatim. If your genetic work can differentiate between kinds and separate humans from chimps, why are so many pairs that you accept as related less similar?
I'm not sure I understand the question.
See, if your genetic work can differentiate between kinds and separate humans from chimps, why are so many pairs you accept as related less similar? Um, I don't understand the question either. Now think about it. I mean that
Yeah,
this is
I don't know how to answer that.
I don't either. This is verbatim. This is what she put in the chat for me to ask. Um, I don't know. I think it might be relating to maybe a fundamental misunderstanding that we think all kinds are exactly alike, or but we young Earth
Yeah,
creatures actually accept the possibility of biodiversity among kinds.
right. And you know what? Every kind of creature is different. There's more variation within some kinds than there are other kinds. Nothing is so, some creatures, there's very little genetic variation between them. Other types of creatures, there's a lot, but yet they don't, interbreed with other fundamentally different kinds of creatures, and they don't evolve into them.
Right.
And so, life across the board is so amazing. It just really is.
Right.
All these genomes and genomic and cellular systems are so mind-bogglingly complex and amazing. Yeah, but yet the critics are basically all nitpickers, and so they want to find some little anomaly and ignore the 99.9% of the data that screams there's a creator. And that's their mindset. Let's nitpick this little thing here and see if we can find something wrong with it, but it has no bearing on the broad picture of things. And so that's really common. I see that across the board. It doesn't matter what I'm talking about. They want to find some little nitpicky little thing and focus on that, but really ignore the bulk of the evidence. And so, like I say, it comes down to your presuppositions. You know, are you
Right.
just dead set? There's no creator. We all evolved from pond scum. If that's your mindset, then you'll say anything or do anything, and you'll ignore the bulk of the evidence. The Bible says that people are without excuse. There's so much evidence out there. Romans 1:20. There's so much evidence out there that basically people are without excuse. And you want to come up with all these little nitpicky, foolish arguments. You're doing it to your own destruction, eternal destruction, I might
Right.
add.
You think that they would target the whole accusation about cherry picking, because that's the main point of all this, that you're cherry picking the data. This method eliminates that cherry picking.
Right.
And you think that they would sit there and just uh, say, no, we're not cherry picking. But instead of doing that, they challenge the method instead of the point of the method, instead of point of the whole argument.
Yeah, and it's they like to come up with these little esoteric arguments that basically your people don't understand anyways, and much less probably them. So yeah, it's yeah,
Right. All right. If you could you summarize and come to the conclusion to your overall case, and then what based on your conclusion of your findings and so forth, and give us your final thoughts.
sure. So like I said, when I got into this whole issue of whether humans and chimps are 98, 99% similar, I didn't know what I'd find. And in fact, the guy who was leading our ministry at the time said, I want an answer in 6 months. And there was actually two of us. There was myself and one other biologist, and the other guy said, it's going to be longer than 6 months. And he was right. He eventually, left and went to work at a Christian university as a professor. But so, anyways, so when I started the whole process, I began looking at the literature, like I said in my talk, and it was just right off the top obvious that they were cherry picking the data. Even in those studies where they compared the human, chimp, and gorilla genomes, the data that they were getting, they were filtering to an extreme level because they wanted these sequences to be nearly identical so that they would, in their phylogeny tree building programs, because if the sequences weren't almost entirely similar, they wouldn't even work well in these programs. And so, and on top of that, these other studies where they were comparing different regions of the human and chimp genomes, I even corresponded with some of the authors of these studies. And they were literally cherry picking the data. And like I say, a lot of it was just because of the limitations they had on the algorithms they were using. These algorithms cannot compare DNA sequences that are so dissimilar that there's nothing to match up. And so I'm not entirely blaming them for being devious and cherry picking, you know what I'm saying? But the fact of the matter is there were limitations to what they were doing. I mean, how do you compare something present in human, missing in chimp, present in chimp, missing in human, or the sequence itself is so dissimilar that the algorithm can't line things up without hitting, a gap that's too large. So a lot of this is, it's just a fundamental problem with the tools that we have. But cherry picking is a fact. And when I looked at all these top papers that were similar, typically they were protein coding regions of genes. So in other words, they were comparing messenger RNAs into DNA sequence. The uracils were converted out so there would be a DNA sequence they could compare. Anyways, it's just yeah, there was cherry picking going on. Some of it was very intentional. Other types of cherry picking was just a limitation based on the tools. But the cherry picking is a fact. And the chimp genome is not completed to a high level of veracity. It still isn't. And in my opinion, the work that was done at the University of Washington in Seattle, where they actually created a de novo assembly of that data to a bioinformatics lab, and they claimed, they put a link in their paper saying you can download the genome, was a dead link. And the link remained dead for months. I don't know how many months, but it was a long time. And I finally said, what's up? This link was supposed to be good when the paper was published. And they sent me an email back saying, oh, we're correcting the sequence. What do you mean you're fixing the sequence? You didn't even sequence it. What are you fixing? And so in my opinion, I don't know actually what they did, but in my opinion, they were probably trying to make it line up more closely with previous versions of the chimp genome that were humanized. Now, that's just a best guess on my part, but why didn't they make that genome immediately available? And what's this whole bit about how we're fixing things? So I don't, who knows what's going on behind the scenes there, but in that 2018 paper.
There's a question that I just popped in my head that I forgot to ask. On the bugs article, you said that bugs agreed with 84% in a blog. Supposedly, he wrote a peer review paper later, changing the number to 96%. Why does bugs change it to 96% later? What was his reasons for that?
Well, the guy that wrote the paper was Joshua Siemens, and he did a PhD with bugs. And Joshua Siemens was creating DNA visualization software. So the software wasn't necessarily intended to do these really genome-wide alignment comparisons necessarily. In other words, the 96% talked about just the regions that were highly similar, that did line up. And someone sent me an email from Richard Buggs. It was very recent. I think it was within the past year, which was after he published that paper, where he claimed in the email that the 84% still stood, that it was still valid. So Joshua Siemens' genome visualization software was not a sophisticated, BLASTN or BLASTX algorithm. It was just visualization software with a very crude brute force algorithm.
Awesome. Okay. And of course,
And I actually talked to Joshua Siemens at some meetings after that was done, and anyways,
right. And of
I
course,
wouldn't put a whole lot of stock in that.
right. And have other evolutionists besides Richard Buggs also acknowledge a lesser percentage that have come up with other numbers that are much less than that? Because a lot of people don't. Of course, obviously, only the published numbers are the only numbers that agree with that gold standard. I know you didn't talk much about that gold standard, but usually I've heard you talk about that gold standard, the one that they came up in the 70s. But has other evolutionists actually come up with those same numbers as well, or is it just Bugs that come up with that? I heard that they're. Well, I'm trying to see if there's.
Most of them will.
Go ahead.
Yeah, most of them will say, with the best local alignments, and the BLASTN is a local alignment algorithm, meaning it only locally aligns areas that it can actually deal with. In other words, it can't jump across these big gaps that are so dissimilar it can't analyze them. So most evolutionists that are working in the field will tell you that the best local alignments they can get are about 96% before the algorithm hits an area that's so different that it can't. So what Richard Buggs did with the information he put up on a blog, he actually published it also on the BioLogos blog, where he was debating with a bunch of evolutionists. And he also put it up on his own personal blog. But what he did was he downloaded the LASTZ, that's another algorithm, alignments from the UCSC genome browser. He downloaded those and he wrote his own Perl script, which he actually sent me a copy of. And the Perl script basically took the data from this global genome alignment that he downloaded and calculated the amount of nucleotides it actually matched. And it was 84%.
Okay.
And based on an email, and he actually did that not only with the most recent version of the chimp genome, but he did it with previous versions as well. So anyways, I don't know if the Logos people have taken that off their website or not, but anyways, it was kind of interesting because the whole thread went kind of dead after that, after he put that up there.
Right.
And I think he even put up a link to his Perl script, where you could download his script and do it yourself. So, and
Right.
anyways,
What would you I mean, we're gonna go here now, and what would be the last, talking to the audience, what would be your advice on this, and any criticism on maybe things that they've heard, and what would be if you're talking to the audience, what would be your message to be left with?
yeah, I would actually I would get my book, that Human Chimp book, because I tackle the whole human chimpanzee DNA similarity from multiple angles. I mean, I don't just talk about the alignment research I did, but I talk about pseudogenes. I did a bunch of projects on those. I talk about junk DNA. I talk about all kinds of issues that are all interconnected. And if you look at the broad picture, it's obvious that humans are distinct, that there's no evolution going on from an ape. In fact, a child can look at a human and a chimp and tell the difference. And so, you know, I mean, there's clear, obvious differences, and there is huge anatomical differences, as well. And we've got a guy that works with us. He's a medical doctor, and he actually talks about all of those, and they're really extreme. And so, yes, there are some similarities, with the overall morphology between humans and apes, but but there's also pretty extreme differences. And yeah, you have to look at the big picture. Don't be a little nitpicker and focus on something that you think, proves you know, your your idea that you want to reject the creator. I mean, look at, actually like I said before, I was not a Christian my whole life. I grew up pretty much just a total pagan. My parents were pretty much raised me in an evolutionary environment, and so it's not like I grew up with all these, creationist presuppositions. I basically cried out and said, God, if you're out there, do something. And sure enough, within a couple of months, and it was honest. I mean, I was honestly saying, Lord, God, if you're out there, if you're real, do something. And he did. But people just, are you at that point where you actually want to know the creator, and or do you just want to just put up this front your whole life and end up in destruction? And so, ultimately that's what it boils down to. So,
Right.
ask God to reveal himself to you, quit all your little nitpicking to support your presuppositions that we evolved from an ape, and or whatever it is that you're holding on to. And so anyways, I guess that would be my recommendation. Just get honest, with your creator. And if you don't want to, then that's your decision.
Right. I also grew up as a evolutionist before I was converted to Christianity, so I have that experience as well. I just high school high school science, I guess, what they call science. Evolution is taught in the public schools and so forth, and so I grew up with that type of mentality, and I love science all throughout my life. So, but and of course I was convinced because all the scientists thought so. So I thought I would, So I agree with evolution all the way up until my conversion. So I come from that background as well. All right,
Well,
this
when yeah. Well, just one more thing, and after I became a Christian, I was in college. I'm getting bombarded, by my professors with all this evolution, and I began studying, the issue for myself. And I didn't know what I'd find, but I was surprised to read all of this literature that pointed out that evolution had major problems. That when you look at science, whether it's geology, paleontology, whatever, it all screams that Genesis is real, and it's getting more so the more we go on here. We have people that work here as physicists and geologists in other fields, and everything they discover is increasingly confirming the truth of Genesis. And so it's even more convincing now than it was back, when I became a Christian back in the 1980s. So,
right,
but even the information they had at that point totally debunked the macro evolutionary paradigm.
right. All right. We're going to end it here. It has been such an honor to talk with you, Dr. Tompkins. Uh, been going on for a little bit. It is great. Your presentation is good. We thank you for taking this time. I'm glad I caught you before you went on your trip this weekend. So I'm glad
Yeah,
I managed to catch you in time. So it's been awesome. Thanks for responding to my email. Honestly, when I sent that email out, I didn't even know you were going to respond back. I just said, well, I'll just take a chance. I just got through doing an interview with a buddy of yours, Dr. Nathaniel Jensen,
yeah.
Evolution. And I just got through doing an interview with him, and we even talked about your research as well a little bit. And I said I want to interview Dr. Tompkins, and I started doing some research and started to backtrack, because you put your information on how the or how you on the parameters you use for your experiments and so forth on the internet. So I started checking out some information. I come across your email, and I was like, well, I'll see if he responds or not. But
Yeah.
it has been awesome. I do appreciate it. I very much appreciate coming on here, and you've answered a lot of questions. I've learned something definitely about your method, about a lot of these criticisms that are going on the internet. So I think that it has been an awesome time. I think that you gave some awesome responses, and I do appreciate it. We all appreciate it. Thank you, Dr. Tompkins.
You're welcome. I appreciate it.
All right. God bless.
God bless you. Thanks.
Yep. Thank you for watching Apologetics 101. It's been exciting. Since I started researching this, I've had critics from all over the place, Gutsy Given, Erika, and others, just attacking and responding to these kinds of arguments and making all kinds of criticisms. I'm so glad that Dr. Jeffrey Tomkins did not disappoint. Let me know in the comments what you think of his arguments. He does a good job responding to the argument, responding to these criticisms, and people that try to replicate his experiment. That's one of the biggest things that's out there now. I can take my personal computer, do it in the next couple hours, and just replicate everything that took him, months, in some cases years to just hone in and be able to actually do an impressive study regarding the human and chimp genome and making comparisons to it. So it has been, and the idea that chimps and humans are related, there's zero evidence to that, and the evolutionists need this. They might deny needing it, but they do need it. That's why they're so big about it. That's why there's lots of criticisms about it. The young Earth paradigm can actually explain both similarity and dissimilarity within the genome. We can explain both biodiversity, and we can also explain similarity. This is squarely explained within the young Earth paradigm. So our views don't actually require any amount of similarity or dissimilarity to be true between an item. There are fundamental differences between kinds, of course, but even in those cases, we still allow biodiversity as long as they don't deviate away from the kind level. And so, but with evolutionists, they require this. They require that the numbers be as high as they could possibly go. And something I've always believed is that the evolutionist was cherry picking that number, cherry picking that data, and it turns out that they were cherry picking it. He pointed out things like they were target, and some things are just like he said, it's just not their fault. It's not their fault that what the algorithm could pick up, but the algorithm will pick up the most similar areas and ignore dissimilars. But then other things are much more in line with what they're choosing to be, so that they can get that high 90 percentile range. And some evolutionists have gotten even lower ranges than that. These usually don't get published in the literature, of course, because they're only willing to publish a high 90-something range. And of course, a lot of skeptics would then pounce on this and say, well, see there, they all got the same. No, there's lots of numbers that didn't get that range at all. The problem is they didn't get published because they didn't give them the range that they want. They didn't give them the high 90 percentile range. They want the high 90 percentile range. They need the high 90 percentile range, because otherwise they have zero evidence for chimp and human relationship. So the question of the hour is, are humans and chimps related? According to evolutionists, they think that they are. I would say no, they are not. They are not related. There's no evidence at all of relationship. They are different kinds of organisms that God created. Trying to make that connection, people have been trying to make that connection since Charles Darwin. And I just don't think that the evidence supports it. I don't think the data supports it. And I think that it is a moot point for evolutionists to keep on trying to kick at that dead horse. It's not moving. It's not going anywhere. So evolutionists that are watching this, sorry, but no, humans and chimps are not related. We're very much different from chimps. We would expect some similarity, in all organisms. There's similarities between us and mice. Doesn't mean we're related to mice. You know, sometimes we accuse men of being mice, some men being mice, but that's not the same as us being related to a mouse. So yeah, I don't see any reason to justify this claim that humans and chimps are related. And people that have tried to, or claimed to have tried to replicate the experiment, I am very doubtful of that and very questionable of that, because essentially, if you actually read the methods in the papers, he uses a lot of advanced equipment, he uses a lot of advanced computers, he uses, they're sequencing genes and chromosomes, and it's taken a long time to do that. And you got people like Gutsy Gibbon, that there are people that think that it took them overnight, hours, a few hours. He mentioned Swami Das, a few hours, taking a few hours doing it, a week. What took him months, and it doesn't make a whole lot of sense. It really makes you question whether they've truly replicated the experiment properly. I know Erika from Gutsy Gibbons is going to be watching this video, and I know she's going to get offended and mad and say, yes, I did replicate it. I replicated exactly what's on that thing. And except if she was doing that thing, she would use the right equipment, she would use the right tools, she would use the right resources. She would use all of these things that they put on that list if she was doing it right. Sequencing genes, looking at it letter by letter, she would do all that stuff. All they basically did was took the numbers that was in the parameters and then try to figure out an average, assume and assumes that he did in this way. He's not using an Excel program to come up with an average like that. So it winds up not being able to make much sense of that you used your home computer at home, you did this in a short period of time, and just simply using BLAST or whatever other algorithm that they can come up with, that's not the same as replicating exactly. I find it very hard to believe that she or anyone else had replicated, including Swami Das, but a few hours, that doesn't make no sense. It takes a long time to actually look through that genetic evidence. And it makes you wonder, if there's deception involved, that sort of things. Like, how in the world do you possibly do that? And I don't know if these people are lying. I don't know if they're telling the truth, but they're sadly mistaken if they think that they can accomplish this successfully in the amount of time that they did it, with the amount of resources, equipment, and computer programming limitations. The fact that they're not actually sequencing the gene, they're not actually going through it, whether instead they're just kind of averaging numbers up there in an Excel program and other kinds of programs. So it really kind of bites the bullet when it comes to trying to figure out whether or not if these people really could have replicated that experiment. I just don't see it happening. Now, if Erika from Gutsy Gibbon and others and Swami Das want to get the same system, the same program, the same thing, and work off their 2018, like you said, sometimes they focus on an earlier program that had a bug in it and things like that. The more recent data, and by the way, this is something I actually thought of recently, even before I had my interview with Dr. Tomkins. I actually thought of this as well. It's like, because Erika from Gutsy Gibbon would talk about like, well, we sequenced these genomes in 2015 paper and that sort of thing. And so, wait a minute, that's not even his most recent study. His most recent study is 2018. Now, granted, she'll probably counter that with saying, well, I did every single one of them, that sort of thing. But the point is that the 2018 is his most recent study. And now every single time he's done it, regardless how he's done it, it's always been the same. And as far as the weight, that's another thing she focuses on a lot. Well, it's unweighted. Well, the algorithm that they use will always be unweighted, but it's not going to really give you a different number like that. Putting in an Excel program don't make you a geneticist. I'm sorry, it doesn't. That sounds like an interesting idea, kind of a cheap way. That sounds like because I'm always trying to find free and cheap ways of doing things. That sounds like something I might do. Home computer, try at home, do my own little home experiment. That sounds like a cool idea when you think about it. But to then match that up with what Tomkins has done, what he's taken months and in some cases years to perfect and get it right, and use the kind of systems that he's used, that kind of algorithm, going as much algorithms, the much data sets that he's gone through, and still be able to accomplish what Tomkins has done. It really just kind of undermines that. It really doesn't take in the value of that research, that study that he's done. It tries to devalue it by making it look like, hey, I can take my own computer and prove you wrong, regardless of how much tools, resources, money, regardless of how much lab equipment you use, regardless how much time you spent on it. I could do this within a few hours on a personal computer. It's really a very insulting, degrading sort of argument. And I would advise anyone to sway from someone who thinks that they can accomplish what Tomkins has done, at least with their limited parameters like that. And most of these skepticism, most of these criticisms, they're coming from the internet. They're coming from non-geneticists. If these criticisms were so impactful, so detrimental, you think that geneticists who are evolutionists would be using these arguments. You think that they would write a peer review article against what Tomkins has done. You think that they would do that, but you don't see them do that. I'm not just talking about in Nance's research journal, even a secular literature like Science or Nature or something of that nature. You could do that, but yet you don't see that floating around. You don't see that being cited against us. Now, some people say, well, they always come on, not only the published numbers come up there, but the point is that the evolutionist who agrees with that high number, the very ones that would have reason to want to challenge Tomkins' methods and would have the resources to challenge his methods, aren't doing it. Why is that? If what Tomkins has done is so wrong, if that's just not the way it should be done, then why aren't we hearing more about it from the evolutionary community that are geneticists? It's just something to leave you with, to think about. But it's been great. I enjoyed my conversation with Dr. Tomkins. I hope you did too. Drop me a comment below. Don't forget to subscribe to my channel if you haven't subscribed already, and like the video. I plan on producing more. I might get into, I know he started talking about Australopithecus, and that's not really the focus of this discussion. But I'm glad he brought it up though, because I do plan on jumping in that subject. It might even be the next one I target. But I plan on going there and getting some experts involved in that. So if you like to see that, don't forget to drop me a comment below. And if you want to see more things on Australopithecus, I'm going to be bringing it in out. But I would love to hear from you. I'd love to hear what you think, your ideas, your suggestions, topic matters you'd like to see me cover. by all means, leave it in the comment below, and I would love to hear from you. We have a lot more videos planned up. Don't miss out a thing. Make sure you subscribe to my channel. Make sure you like the videos. Make sure you hit that notification bell so you can be alerted whenever I produce a new video. I can't wait for who I'm going to interview next. It's great. Dr. Tompkins was the first one I've ever interviewed from Institute for Christian Research. Hopefully he won't be the last. I would love to interview a few more from there. And so we'll see what happens. But it's been great. And I will see you on the next one. Bye.